Hello, everyone. Welcome back to the channel. This is Dr. Hasi, and today's topic is chi-square test, Mendel-Genetics, and Mitosis and Meiosis. So 3 chapters, so we are going to review today. So first, we start with chi-square analysis or chi-square test. So in chi-square test, we have basically we start with a question, which you are given a question usually. So question might be asking you, does this data fit to 3:1 ratio? If that's the question, you have 5 steps. Step number 1, you are going to write an hypothesis, which is HO. So your hypothesis will be data follows 3:1 ratio. You just state the question as a statement. Then and then you are going to actually have a blank table, which you will have these 1, 2, 3, 4, 5, 6 columns in the table. And the first column is always phenotype. So 3:1 in this case, maybe 3 green to 1 white. So your phenotype is 3 out of 4 green, and that's first row. Second row, 1 out of 4 white. So you put them in first column. Second column always given to you, observed numbers. So in this case, our numbers is 824 and 297. And then third column is expected. You need to calculate this. Then the other ones, next column is observed minus expected. Then observed minus expected square. Then finally observed minus expected square divided by expected. So always remember, when you add these column number 2 totals and column number 3 totals, they should add same number. If you add these two, if this is 1000, and the column number 3 also should add to the 1000. So how can you find expected? That's the first column you need to calculate. Expected equals total, like which these two added, 824 plus 297, 1000. And if you add them together, that total multiply by the ratio, which is 3 out of 4 here, and that will give you a expected number, which is 840. You do the same thing, total times 1 out of 4, and then you find the expected number for second row. And then the rest is easy. You observe minus expected, this number, 824 minus 840, 297 minus 280, and then you end up finding this last column and add the totals, and then this total is 1.34. This is called chi-square calculated number. We need this number because we need to use it in next step, which is step number 3, chi-square table value. Chi-square table value, a lot of time we use is 3.84. So we got it from the chi-square table using degrees of freedom, df, which is equals n minus 1. N is number of phenotypes, 2 minus 1, 1. We go to column 1 and then find 3.84. You don't need to memorize this number. Then the step number 4, now we know chi-square cal and chi-square table. We compare them. We always want our chi-square calculated number we calculated from the table smaller than chi-square table value in order to accept our hypothesis. Is it the case? 134 is smaller than 3.84. So our result, which is step 4, yes, we accept HO or our hypothesis. And a conclusion, step number 6, is step number 5, which is conclusion. Since we accept it, we go and read our hypothesis again, which was data follows 3:1 ratio. Our conclusion is, yes, data follows 3:1 ratio. This is chi-square analysis with 5 steps. So if you have questions, please let me know. In the class today also, we are gonna also review and answer any questions on that. Okay, so this was one of the labs, chi-square test, that we use growing corn plants and then testing them for, like, let's say 3:1 ratio or 1:1 ratio. So next is Mendelian genetics. Since we talk about ratios, Mendel, basically, this is the lab number 13 or chapter lab 13 in your book, Mendelian Genetics. Always remember, we should always start with Mendelian Genetics with the definitions, the vocabulary. Okay, so genetics always start with vocabulary, right? So basic vocabulary. So you see here, I have a list of them. Always remember phenotype equals genotype times environment. Phenotype can be wild type or a mutant. Genotype can be homozygous, heterozygous. We use letters like A, B, C, D, E, F, G, K, L, any letter that's actually in the alphabet pretty much for naming genes. The only thing you need to pay attention is large letter is dominant, like big letter, big A is dominant, but small a is recessive, right? So you need to pay attention to this. Now, okay, so some other vocabulary like gametes, you know, from the meiosis, mitosis, which basically important here in genetics. There are different levels of level of generations. We start with in plant genetics, Mendel-Genetics. We start with parental generation, P1, cross it with P2, and then we find the gamete from meiosis. If we have big A, your gamete is big A. If you have small a genotype, you have your gamete is small a. But if you have a heterozygous genotype like big A, little a, then you have two gametes. They are big A and little a. You need to pay attention. So you should always be able to go from genotype to gamete, gamete to genotype, both direction, right? So make sure that you pay attention to those. And haploid means just one set. Diploid means two sets. Triploid means three sets. It goes on like that. So crossing over is basically what Mendel did in Mendel's experiments. So you need to follow the experiments. If you cross two parents, go to F1. If you cross F1 and self it, you get F2. We always look at F2 ratios, that what Mendel did. So first ratio was 3:1 ratio, 3 dominant, 1 recessive for green and white flower pea plants. But he did 8,000, he grew 8,000 plants, and then he did many experiments. He also looked at 2 gene problems. So 3:1 ratio is 1 gene problem. But he also looked at 2 gene problems like seed color and seed shape. And then his F2 ratio was 9:3 to 3:1. So also remember that. You might look at some other things like phenotype is physical appearance, as I already mentioned, equals to genotype, which is genetic makeup, times environment. Okay, so genotype is all about genetics. Environment is more abiotic, more non-living things that might affect the gene expression. Okay, there is also something called test cross. If you want to find the unknown phenotype, that you can actually do a test cross and then look at it. And then if you can look at your possibilities. One probability is you get 1:1 ratio. The other one is 1:0. So you can actually guess the phenotype of unknown parent. So look at the definitions, go over each the problems in the book. Basically, it says genetic problems, page 87. So always remember, dominant doesn't mean always fully dominant. There might be complete dominance like Mendel did. Incomplete dominance, we see sometimes like snapdragon plants that they don't completely dominate each other. And then there is another dominance, which is big genetic things, which is blood types that we get genetically. If you have type A in one parent, type B in another parent, we get actually AB, which is called co-dominant. So pay attention to those. And let me know if you have any questions on this. The last chapter was meiosis and mitosis. So mitosis and meiosis. This is something to do with cell cycle. So remember, there is cell cycle. The cell cycle goes interphase, mitosis, and cytokinesis. In most cells go through a regular cell cycle, which is basically mitosis, but gametes, the special cells, they go through meiosis, which is also we seen in the following chapter here. So mitosis has this 4 steps plus interphase. Meiosis has 10 steps. You need to be familiar with both mitosis and meiosis steps, and then make sure that you can identify each step. So let's go over and review in both mitosis and meiosis. So interphase goes G1 S G2, right? So remember, remember this cell cycle. Interphase is where we start, G1 S G2. Then we start with the phases of 4 phases of mitosis: prophase, metaphase, anaphase, telophase. You should pay attention how chromosomes look like. In interphase, we don't see them under the microscope. They look cloudy, but in prophase, they show up. They are duplicated. In metaphase, they are lined up in the middle. Anaphase, they split and go in the 2 opposite directions. Telophase, they already move to the 2 opposite directions, and there are 2 nucleus, and that's end of mitosis. And then next is cytokinesis, which is division of the cytoplasm. So mitosis is pretty straightforward. Again, most cells go through mitosis using repairing cells, growth, and replacement. So mitosis basically produces exact same replica of the parent cell, makes 2 copies exactly same. Meiosis, M-E-I-O-S-I-S, which is the 10 steps, this long process, right? So meiosis, the meiosis basically, it is like 2 mitosis back to back, right? So it's still, there is interphase, but there is only 1 interphase. If we start with 1 cell, we end up with 4 daughter cells. They are actually half the chromosome number of our parent cell. Unlike mitosis, meiosis half the chromosome number. There are 10 steps of meiosis, and it's like prophase, metaphase, prophase 1, metaphase 1, and anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So everything doubled except interphase. Okay, so the end products are 4 haploid daughter cells. If we start with 1 diploid parent cell, we end up with 4 haploid daughter cells. So always remember. So meiosis, the main product is production of gametes, like egg or sperm cells. So always remember, there are 2 cell divisions, meiosis 1 and meiosis 2, 4 daughter cells. Each of these 4 daughter cells are not same. They are just unique to each other, and then they form egg and sperm cells. So keep working on these 3 chapters, and I will see you in the class. We will continue reviewing on this in a minute. So thank you for watching, and please feel free to comment and subscribe. Happy studying. See you in the next video.